Hello and welcome to today's video. It is on diversification and speciation. And from QCAA, here's our learning goals. Firstly, to identify that diversification between species can follow one of four patterns: divergent, convergent, parallel, and coevolution. Secondly, to describe the modes of speciation, that is allopatric, sympatric, and parapatric. And thirdly, to interpret gene flow and allele frequency data from different populations in order to determine speciation. So you can pause the video here and maybe just write your own version of those learning goals down. Cool, let's get into it then. So first up, types of evolution, and we have down the side here just a bunch of key terms taken from your textbook. And I would suggest to write those down in the glossary that you guys are definitely developing too. So first up, to identify that diversification between species can follow those four patterns we said. What does it mean to identify? Well, it means to distinguish, to locate, recognize, and name. To establish or indicate who or what someone or something is, to provide an answer from a number of possibilities, and to recognize and state a distinguishing factor or feature. So basically, it means to identify something and maybe provide a little bit of evidence as to why it's that. So let's get into it then. Divergent evolution. It refers to two or more distinct species which share a common ancestor from which they both diverged, as the name suggests. And a common example is the various dog species that diverged from the common ancestor, being the common grey wolf. They all evolved independently, but from that same ancestor. Convergent evolution, though, it refers to two or more species which share traits but not due to a common ancestor. So the example of this being birds, bats, and butterflies. They all have wings that allow them to fly, but they do not share a common ancestor with wings. So each one evolved to fly independently. We can say that evolution converged on a similar trait in vastly different species. So that's convergent, that's divergent. Parallel evolution occurs when two species that share a common trait due to a common ancestor evolve similar traits but independent of one another. That is often due to similar environmental pressures, and a common example of parallel evolution is the marsupial mammals of Australia. So here we have the little sugar glider, but also the placental mammals of elsewhere in the world. So these mammals are said to have once shared a common ancestor, but were separated early on, but despite the separation evolved many of the same traits. And in this case, number one being similar gliding traits and abilities. So if we just think about those three that we've covered. To diverge means they came from a common ancestor and diverged. To converge means they came from different ancestors, but the traits are becoming more similar. Whereas parallel means they have similar traits, but they're evolving fully independently of one another. Which leads me to the last one, and that is coevolution. Coevolution occurs when two or more species reciprocally affect each other's evolution. That is when two species head down an evolutionary pathway together. So a great example being the insects and the flowers. This bee, for example, is taking nectar from the flowers and in turn transferring some of this pollen that it lands on from the flower to another flower, and the insect and flower are both getting an advantage from that. So it has worked out so well, this relationship, that half of all the animals on the planet are in fact insects, and flowering plants which are called angiosperms, make up most of the plants that we see around us, and that even includes the grasses and the big kind of trees around us. Both have benefited from this symbiotically mutualistic relationship. So in summary, we have divergent evolution. One common ancestor, parent species, from which they both diverged, such as the wolf and the dogs. But convergent evolution is when we have this kind of separate parent species, but they come together with a similar trait. And that could be like the flying ones. Whereas C is parallel evolution, and that occurs when we have, maybe they once started off from one common species, but anyway, for whatever reason, they're separated and they develop these similar phenotypes and genotypes based on maybe being in similar environments. So they're said to be parallel. And that is types of evolution. So let's keep going. Isolation and speciation. The learning goal was to describe the three types. What does it mean to describe? It means to give an account, written or spoken, of a situation, event, pattern or process, or of the characteristics or features of something. And we know that defines generally looking for about 1 to 2 sentences and describe about 3 to 4 worth of key points and information. So, allopatric, sympatric, and parapatric. They are not all the types of speciation. There's also peripatric, which is P-E-R-I, but that's beyond the syllabus, so we're just going to focus on these three. With these, I'm going to show you a diagram here, and there we have the three types and kind of how we get speciation. So speciation just means a new species being formed, and there's three main ways that it can happen. If we looked at this, we can see that, well, it all starts off the same and all ends the same, but the way in which it gets there varies. So this is showing that we have this one population, but by the end of this process, we have two populations. So if we think about how it starts off with one and ends with two, well, we know that a species means that they may be similar. We could call them sister species, but they do not interbreed with one another. So no interbreeding can produce viable offspring anyway. I should clarify. They may be able to interbreed, but can the offspring interbreed successfully or not? So now let's look at each one more closely, and we'll start off with allopatric speciation. And here there is always an ecological or an evolutionary event. So that could be a physical barrier that forms, and it's usually geographical. We will look at some contextual examples after these theoretical explanations. But there's a physical barrier, and that separates the two, and in doing so, maybe one retains its form, but the other adapts to its new environment. And this gives opportunity for two populations to enter what is called reproductive isolation from each other. And in doing so, results in two new species. So reproductive isolation, as the name suggests, they just don't interbreed. And why? Well, in this case, there's a physical barrier, physically stopping them from doing so. Last week we spoke about phenotypic selection pressures, and I'm going to tie this kind of content that we're covering this week back into that. So generally, allopatric speciation results in reproductive isolation, physical isolation, and this is generally associated with disruptive selection. So if you think back to disruptive selection, that was when there was this selection pressure in the middle. And that was the rodent example that we used last week, the really stumpy tails and the really long ones. That's allopatric. Parapatric speciation is different though. It's when we have a situation where a subset of the population enters a new niche. And this may be because a new niche opened up for whatever reason. Maybe one species was outcompeted. Maybe there was an extinction event that happened. And in doing so, they form what is called niche isolation. And this allows a subset of the population to thrive and to speciate into a new form. This favors what we call and remember as directional selection. Directional selection being the species literally shifts its phenotype to utilize the new resource. Thirdly, sympatric speciation, as the name suggests, is where you have speciation that occurs very distinctly in a fixed geographical space. Sympatric literally means like in the same home. The only way this occurs without a new niche opening up is by a mutation occurring in the population, which confers the slight selective advantage for the species. There's no change in the environment in sympatric speciation as there was in the other two. It's a change in the population, and we call this genetic polymorphism. And an example could be just perhaps it's a better way of using an existing resource. Maybe it's a better way to detoxify a plant for consumption. Either way, this results in local isolation. So isolation within the species of that subset within the greater population, again leading to two different species. And this is associated with stabilizing selection. So in summary, there are three main types of speciation and how they integrate back into the idea of phenotypic selection from last week. So how do we describe this as the subject matter expects us to? Well, the implications from the questions is to describe population units, and that being the original population being a single unit and the resultant population being two separate units. We can talk about gene flow, whether there is still the opportunity for gene flow to occur as they could be here and as they could be here, but definitely not here where there is a physical barrier stopping them. And then we can talk about the resultant species as a result of these processes occurring. And lastly, if we were to consider some contextual examples, today we see two separate squirrel species either side of the Grand Canyon, which were believed to once be one species, physically divided when the canyon was formed. And this is, of course, physical barrier, an example of allopatric speciation. Parapatric speciation can occur sometimes when polluted environments, for example, high levels of minerals such as iron and zinc can prevent plants from growing, but some plants such as buffalo grass can actually really utilize that niche as they have the ability to live and grow and detoxify what would probably kill off most other species of plant. So that would be utilizing a new niche isolation, parapatric. Whereas sympatric speciation is more common in plants than it is animals. An example being the maggot fruit fly. So these flies only mated on a particular tree. They were hawthorn trees. But when apple trees were introduced to America, so a couple of centuries ago, they began to mate on these apple trees as well. However, a funny thing kind of happened where we saw this splitting between the flies, because like the flies that were conceived on the apple trees, they would only want to mate on the apple trees as well. But the flies that were conceived on the hawthorn trees only wanted to mate on the hawthorn trees as well. And there began this splitting that we're starting and continuing to observe just over a couple of centuries. So that's an example of sympatric within the same home isolation. Okay. And that is all for as far as the theory and content goes. We have allopatric speciation, parapatric, and sympatric. So let's get into some questions now. First up, multiple choice, and it's asking us to choose which definition best suits allopatric speciation. So I'll put this image up here too. And you can pause the video here and see how you go. Okay, hopefully you guys have been able to work through this. So I think, all right, here we go. A, is it when two subpopulations, that's boom, boom, or dun, dun, or dun, dun, become isolated from each other, which prevents genetic exchange? Allopatric. Sounds pretty good. Or is it when two subpopulations of a species evolving in reproductive isolation while continuing genetic exchange? Well, we know allopatric has a physical barrier that causes reproductive isolation, so it can't be B. Is it C, when new species evolving from a single ancestral species while inhabiting the same geographical location? That sounds like kind of sympatric to me. They're definitely two distinct areas here. Or is it a new species forming from an isolated peripheral population? That sounds like parapatric to me, so the answer is D. Oh, sorry, the answer is A. A. Okay, next question. Question 1. It is a short response, and it's asking us to describe the process of parapatric speciation. So I'll just put that there to jot your memory, and when we're looking for 3 marks, kind of like 3 key points. The QCAA exemplar has a couple of terms that we haven't covered in this video, but we can make a little bit of sense of it when it comes up. But do your best, go for it, and we'll see how you go. Okay, hopefully you guys have come up with a smashing answer. This is what QCAA says. So you get a mark for saying there's 2 subpopulations where there's reproductive isolation, and you get a mark for saying there may be some kind of mating that occurs here, but it's pretty random and it's not continuous, it's not even. In fact, it's so not even that, like, this is a smaller population, and we know small populations are more susceptible to genetic drift, thinking back. So here is their response. In parapatric speciation, 2 subpopulations of a species evolve reproductive isolation from each other, but continue to exchange genes. There is no, why? Because there's no specific barrier to stop that gene flow like there would be in allopatric. So the population is continuous. It can still kind of continue, but has random mating. A smaller population is isolated, usually at the periphery of a larger group, with unequal gene flow remaining between the 2 populations. Cool. So maybe you can check this out a little bit more if you want to go in depth, but that's that. That was all there was in the mock stimulus for this topic. So I've put in another question here, and it is to compare sympatric and allopatric speciation in terms of these 3 kind of criteria. If you think back to Dr. G when he did the experience genetics kind of online excursion for you guys, he talked about to compare means to display recognition of, yeah, similarities and differences, but to go further, hey, and to say the significance of those. So you can use your textbook. It goes into a bit of detail with this. Otherwise, you can use your knowledge from this video. By the way, pause the video and give that a go. Cool. All right, let's keep going. Last question. Do you remember that final QCAA learning guide said to interpret gene flow and allele frequency data? This is something we've kind of been doing for a couple of weeks, but let's do another question. Question 28. Maybe you remember this back from term 1 when I gave you this way too early. So the question shows gene flow between 5 different populations in hypothetical species. The arrows represent the gene flow direction, and the size of the circle represents the size of the population. And an event that causes R to become geographically isolated. Okay, so the questions. A, 4 marks. Obviously there's way more lines, but I just cropped it for the video. Identify which population or multiple populations are most likely going to form new species, and to justify your answer, I'll give you B as well. It says to identify which population would gain a selective advantage if an environmental selective pressure that favors phenotypic diversity is applied to this situation to justify your answer. So pause the video here and see how you go. Okay, so this is a big one. This is worth 7 multiple choice questions that you get correct if you do this one. Let me show you what QCAA did. Alright, so 4 marks for A. Identifies that M could form a new species, 1 mark, and R could form a new species, and provides a valid reason for each of those. So if we think about which is more likely to form a new species, well, firstly, there's 2 kind of parts here that we could pull upon. Maybe I'll just read this to start with. Population M and R could form a new species because they would be isolated. So that's identifying, 2 marks there if you got M and R. Why? Let's get those other 2 marks. The effect of genetic drift is strongest in smaller populations, and these 2 are the smaller populations. Why? Because they're just way more susceptible to influx and outflux of genetic information. So there's one reason. So both populations, M and R, are more likely to be affected by this, creating divergence and possible speciation. So they used size for this. We could also kind of say, well, T looks super safe. It's got genes going in and out. It's a big population of whatever species. D only has genes going out. So that would be the next pick, but clearly M only has genes going out, and R is the next smaller one. So mainly the size. That's where the most logical answer is there. B talks about which one is gain a selective advantage. So if you said T, it would gain a selective advantage if an environmental pressure came into exist. Why? Because well, it has flow going in and out, and also it's just the biggest population, so it's got the higher probability of survival and reproduction. So if there was like a selective pressure that was applied, it's most likely to maintain its diversity. And that is that question. Let's finish up with a couple of quotes. And so here is a quote, and it says, new mutations don't create new species, they create offspring that are impaired. Perhaps you think, oh, okay, this is another Christian quote that Mr. Callan's given me. However, it was by this lady called Lynn Margulis. I hope I pronounced her name right. And she was actually a religious agnostic and staunch evolutionist. So she didn't believe in God at all, that we know of. And that means, yeah, she was coming from a scientific point of view here. She was an esteemed and renowned biologist, even admired by other similarly notable big figures such as Richard Dawkins. And this is her. And I put her quote here though to show you that like the perspectives that we read about in the textbooks, although they're spoken like gospel, they are even challenged by well-known scientists in the evolutionary field. She actually stated a really different kind of idea to how we get all this diversity and variation, and it was like through this process of symbiosis with viruses and bacteria exchanging genetic material. But I just want to show you that to show, like, even though what we read, it looks like really dead set, it's actually not bulletproof. There's a lot that we could say about other perspectives. And I'll even give you this other quote that she said. She says, natural selection eliminates and maybe maintains, but it doesn't create. It sounds similar, hey, to alternative perspectives we've talked about. Neo-Darwinists, so people who believe in Darwinian evolution but use genetics to support it, say that new species emerge when mutations occur and modify an organism. And I was taught over and over again that the accumulation of random mutations led to evolutionary change, which we read about in textbook, which led to new species. And I believed it until I looked for evidence. So there's quote 2. And to finish, here's another quote. That natural selection can produce changes within a type is disputed by no one, not even the staunchest creationist, but that it can transform one species into another. Well, that in fact has never been observed, and that is by this man, Robert J. Sawyer. He's an author. And I just present you this information on this slide and in all these videos and these quotes in hope that you will continue to do your own research and to weigh up the evidence that we look at and talk about in class for yourself, so that you may come to your own place of understanding and belief, in hope that you will not only know what you believe, but why you believe it, and likewise what you don't believe and why you don't believe it. Anyway, I hope that this video has been informative, and I look forward to seeing you guys in class. God bless.